We have a global health challenge in our hands today, and that is that the way we currently discover and develop new drugs is too costly, takes far too long, and it fails more often than it succeeds. It really just isn't working. And that means that patients that badly need new therapies are not getting them, and diseases are going untreated. We seem to be spending more and more money. So for every $1 billion we spend in R&D, we're getting less drugs approved into the market. More money, less drugs. So what's going on here? Well, there's a multitude of factors at play, but I think one of the key factors is that the tools that we currently have available to test whether a drug is going to work, whether it has efficacy, or whether it's going to be safe before we get it into human clinical trials are failing us. They're not predicting what's going to happen in humans. And we have two main tools available at our disposal. They are cells in dishes and animal testing. Now let's talk about the first one. Cells in dishes. So, cells are happily functioning in our bodies. We take them and rip them out of their native environment, throw them in one of these dishes, and expect them to work. Guess what? They don't. They don't like that environment because it's nothing like what they have in the body. What about animal testing? Well, animals do and can provide extremely useful information. They teach us about what happens in the complex organism. We learn more about the biology itself. However, more often than not, animal models fail to predict what will happen in humans when they're treated with a particular drug. So we need better tools. We need human cells, but we need to find a way to keep them happy outside the body. Our bodies are dynamic environments. We're in constant motion. Our cells experience that. They're in dynamic environments in our body. They're under constant mechanical forces. So, if we want to make cells happy outside our bodies, we need to become cell architects. We need to design, build, and engineer a home away from home for the cells. And at the Vis Institute, we've done just that. We call it an organ on a chip. And I have one right here. It's beautiful, isn't it? But it's pretty incredible. Right here in my hand is a breathing, living human lung on a chip. And it's not just beautiful. It can do a tremendous amount of things. We have living cells in that little chip. Cells that are in dynamic environment with interacting with different cell types. There's been many people trying to grow cells in the lab. They've tried many different approaches. They've even tried to grow little mini organs in the lab. We're not trying to do that here. We're simply trying to recreate, in this tiny chip, the smallest functional unit that represents the biochemistry, the function, and the mechanical strain that the cells experience in our bodies. So how does it work? Let me show you. We use techniques from the computer chip manufacturing industry to make these structures at a scale relevant to both the cells and their environment. We have 3 fluidic channels. In the center, we have a porous, flexible membrane on which we can add human cells from, say, our lungs. And then underneath, they had capillary cells, the cells in our blood vessels. And we can then apply mechanical forces to the chip that stretch and contract the membrane. So the cells experience the same mechanical forces that they did when we breathe, and how they experience them, how they did in the body. There's air flowing through the top channel, and then we throw a liquid that contains nutrients through the blood channel. Now the chip is really beautiful, but what can we do with it? We can get incredible functionality inside these little chips. Let me show you. We could, for example, mimic infection, where we add bacterial cells into the lung. Then we can add human white blood cells. White blood cells are our body's defense against bacterial invaders. And when they sense this inflammation due to infection, they will enter from the blood into the lung and engulf the bacteria. Well, now you're going to see this happening live in an actual human lung on a chip. We've labeled the white blood cells so you can see them flowing through, and when they detect that infection, they begin to stick. They stick, and then they try to go into the lung side from the blood channel. And you can see here, we can actually visualize a single white blood cell. It sticks, it wiggles its way through between the cell layers, through the pore, comes out on the other side of the membrane, and right there, it's going to engulf the bacteria labeled in green. In that tiny chip, you just witnessed one of the most fundamental responses our body has to an infection. It's the way we respond to an immune response. It's pretty exciting. Now, I want to share this picture with you, not just because it's so beautiful, but because it tells us an enormous amount of information about what the cells are doing within the chips. It tells us that these cells from the small airways in our lungs actually have these hair-like structures that you would expect to see in the lung. These structures are called cilia, and they actually move the mucus out of the lung. Yeah, mucus, yuck. But mucus is actually very important. Mucus traps particulates, viruses, potential allergens, and these little cilia move and clear the mucus out. When they get damaged, say by cigarette smoke, for example, they don't work properly and they can't clear that mucus out. And that can lead to diseases such as bronchitis. Cilia and the clearance of mucus are also involved in awful diseases like cystic fibrosis. But now, with the functionality that we get in these chips, we can begin to look for potential new treatments. We didn't stop with a lung on a chip. We have a gut on a chip. You can see one right here. And we've put intestinal human cells in our gut on a chip, and they're on the constant peristaltic motion. There's trickling flow through the cells, and we can mimic many of the functions that you actually would expect to see in the human intestine. Now we can begin to create models of diseases such as irritable bowel syndrome. This is a disease that affects a large number of individuals. It's really debilitating, and there aren't really many good treatments for it. Now, we have a whole pipeline of different organ chips that we are currently working on in our labs. Now, the true power of this technology, however, really comes from the fact that we can fluidically link them. There's fluid flowing across the cells, so we can begin to interconnect multiple different chips together to form what we call a virtual human on a chip. Now we're really getting excited. So now we're not going to ever recreate a whole human in these chips, but what our goal is to be able to recreate sufficient functionality so that we can make better predictions of what's going to happen in humans. For example, now we can begin to explore what happens when we put a drug like an aerosol drug. Those of you like me who have asthma, when you take your inhaler, we can explore how that drug comes into your lungs, how it enters the body, how it might affect, say, your heart. Does it change the beating of your heart? Does it have a toxicity? Does it get cleared by the liver? Is it metabolized in the liver? Is it excreted in your kidneys? We can begin to study the dynamic response of the body to a drug. This could really revolutionize and be a game changer for not only the pharmaceutical industry, but a whole host of different industries, including the cosmetics industry. We can potentially use the skin on a chip that we're currently developing on the lab to test whether the ingredients in those products that you're using are actually safe to put on your skin without the need for animal testing. We could test the safety of chemicals that we are exposed to on a daily basis in our environment, such as chemicals in regular household cleaners. We could also use the organs on chips for applications in bioterrorism or radiation exposure. We could use them to learn more about diseases such as Ebola or other deadly diseases such as SARS. Organs and chips could also change with the way we do clinical trials in the future. Right now, the average participant in a clinical trial is that, average. Tends to be middle-aged, tends to be female. You won't find many clinical trials in which children are involved. Yet, every day, we give children medications, and the only safety data we have on that drug is one that we obtain from adults. Children are not adults. They may not respond in the same way adults do. There are other things like genetic differences in populations that may lead to at-risk populations that are at risk of having an adverse drug reaction. Now imagine if we could take cells from all those different populations, put them on chips and create populations on a chip. This could really change the way we do clinical trials. And this is the team and the people that are doing this. We have engineers, we have cell biologists, we have clinicians, all working together. We're really seeing something quite incredible at the Wyss Institute. It's really a convergence of disciplines where biology is influencing the way we design, the way we engineer, the way we build. It's pretty exciting. We're establishing important industry collaborations, such as the one we have with a company that has expertise in large-scale digital manufacturing. They're going to help us make, instead of one of these, millions of these chips so that we can get them into the hands of as many researchers as possible. And this is key to the potential of that technology. Now let me show you our instrument. This is an instrument that our engineers are actually prototyping right now in the lab. And this instrument is going to give us the engineering controls that we're going to require in order to link 10 or more organ chips together. But it does something else that's very important. It creates an easy user interface. So a cell biologist like me can come in, take a chip, put it in a cartridge like the prototype you see there, put the cartridge into the machine just like you would a CD, and away you go. Plug and play. Easy. Now, let's imagine a little bit what the future might look like if I could take your stem cells and put them on a chip, or your stem cells and put them on a chip. It would be a personalized chip just for you. Now, all of us in here are individuals, and those individual differences mean that we could react very differently and sometimes in unpredictable ways to drugs. I myself, a couple of years back, had a really bad headache, just couldn't shake it. Thought, oh, I'll try something different. I took some Advil. 15 minutes later, I was on my way to the emergency room with a full-blown asthma attack. Now, obviously, it wasn't fatal, but unfortunately, some of these adverse drug reactions can be fatal. So how do we prevent them? Well, we could imagine one day having Geraldine on a chip, having Danielle on a chip, having you on a chip, personalized medicine. Thank you.